discuss. Okay, this week's thought points are the carbohydrates we'll be talking about. As we all know that they are the major components in plant tissues and comprise about 70% or more of the dry matter of forages. So we all know that the chloroplast in the plant tissues, in the plant leaves, synthesize the carbohydrates by means of solar energy, carbon dioxide, and water, and they give off the oxygen. So this is a vital process for animals because they could not exist without this transformation of energy. So carbohydrates consist mainly of glucose and glycogen and make up less than 1% of the weight of an animal. We will be discussing about the functions, deficiency symptoms, and toxicity of certain macrominerals in our second chapter. And then we will also discuss about the mineral toxicities. And sorry about that in the background, my 3-year-old, she is uh, we'll be talking about the mineral toxicities and organic toxins in the food chain. So starting with the carbohydrates. So what actually carbohydrate means is that it may be defined as a polyhydroxyaldehyde or ketone or acids in their derivatives or compounds that yield these derivatives on hydrolysis. The carbohydrates are the neutral chemical compounds that contain the element, as the name implies, carbon, hydrogen, and oxygen, with the last two elements present in the same proportion as in water mostly, but not at all. It has been found that the acetic acid and lactic acid that can be present as hydrates of carbon but are not carbohydrates. The carbohydrates serve as both structural and reserve material in the plant. The animal body contains less than 1% of carbohydrate, which are present in blood, muscle, and liver. So talking about the classification and structure of the carbohydrates, one method of classifying the carbohydrates, you can see there is a table 7.1 in the book. So that's one method of classifying the carbohydrates, like monosaccharides, we have the pentoses, hexoses, and then we have disaccharides, we have trisaccharides, that includes the raffinose, we have polysaccharides, that includes the xylene, hexosins, and glycogen. So that's one kind of classification. Thus, a monosaccharide has only one molecule of sugar. As you can see here, the disaccharide, as the name implies, it has two molecules, and an oligosaccharide may have 3 to 10 sugar units, and a polygosaccharide has more than 10 sugar units in it. Chemically, the carbohydrates are polyhydroxyl aldehydes, as we already talked about, and ketones. So they might have an aldehyde group or ketone group. Aldehyde group, you all know that it's the CHO group, and ketone group means 3 molecules of 3 atoms of carbon and with the double bond with oxygen. So that's the ketone group. So talking about the functions of the carbohydrates in animal nutrition, they serve primarily as a source of energy for normal life processes. However, in plants, some of the simple sugars, especially glucose and ribose, they are involved in energy transformations and tissue synthesis. Some less soluble forms, such as starch, they serve as energy reserves in the roots, tubers, and seeds. The relatively insoluble fractions that include the cellulose and hemicellulose, they are most important in providing structural support for the living plants. You can see here in the picture a class of complex carbohydrates known as glycans. They form an intricate sugar coat surrounding and occupying space between the cells in virtually all living organisms. They have an important role in transmitting signals into and between the cells because they are synthesized in the body. They are not nutrients, so they are not discussed further in this chapter. The broad array of simple carbohydrates found in a variety of foods is shown in table 7.1. You can see here that table in your book. So talking about the functions of carbohydrates, they usually serve as a major source of energy in plant body. They are essential components of production, temperature control, and proper functioning of different parts of the body. They are helpful in absorption of calcium and phosphorus in younger animals and also provide the suitable environment for the growth of rumen bacteria and protozoa. They also maintain the glucose level of plasma and are also components of several important biochemical compounds such as nucleic acids, coenzymes, and blood group substances. The carbohydrates play an important role in the metabolism of amino acids and fatty acids. So talking about the carbohydrates functions, the carbohydrates and lipids are the two major sources of energy for the animal body. It has been found that the lipid content of most diets of food producing animals is less than 5% because most of the energy that's stored in the body, it's in the form of fat. We all know that. So dietary carbohydrate not broken down immediately for energy is stored in the body as fat. So let's now talk about the metabolism of the carbohydrates. So it starts with the preparation for absorption, that's the digestion in the small intestine. Here you can see the structure of cellulose and pectins in this figure. So soluble components of the plant cell wall include the starches, hemicellulose, and beta-glucans, which are not linked to lignin and are available to fermentation. So these starches, hemicellulose, and beta-glucans, they have all the alpha 1,4 linkage in. However, you can see here the glucose, the cellulose, which has beta 1,4 linkage. You can see here in the figure, and is insoluble and resistant to breakdown except by the cellulase enzyme that's produced by the microbes or microorganisms in the rumen and small intestine tract of non-ruminants. So microbial fermentation of cellulose and other plant fibers is that in ruminants, it has been found that in ruminants and other species with large microbial populations in the gastrointestinal tract, there occurs anaerobic fermentation of carbohydrates, which results in the production of large quantities of VFAs, that's the volatile fatty acids. And these include the acid, acetic acid, propionic acid, butyric acid. So these are the three volatile fatty acids that are produced because of the microbial fermentation of cellulose and other plant fibers. So the pathway of this carbohydrate metabolism in the rumen and in lower intestinal tract of non-ruminants is shown in the figure, that how the carbohydrate metabolism takes place in the rumen and in the lower intestinal tract of non-ruminants. So it has been found that the upper or proximal section of the small intestine, duodenum and jejunum, has the greatest capacity to absorb the monosaccharides. The lower, or we can say the distal small intestine or lower ileum, it absorbs less, and the stomach and large intestine absorb the little, if any sugars are present. So how this glucose is transported across the cell membranes, there are two mechanisms for that. The first family that's involved in that is the sodium glucose co-transporters. So we can call them SGLTI. And also we have one more family that's involved in this transmission of glucose is the GLUTI. That's the glucose transformation. That's the glucose uh, transporters that are involved in this transportation. So the second family that facilitates the glucose transporters or the transport the glucose actively down a concentration gradient. So they might be present in all mammalian and probably in all vertebrate cells. The conversion of some monosaccharides to glucose occurs within the intestinal mucosal cells. Like here in the figure, you can see the conversion of fructose to glucose remains relatively constant over a wide range of fructose concentrations. But the rate of movement of fructose into the cell is roughly proportional to the luminal concentration. It has been found that the fructose is converted to lactic acid by the intestine of some animal species. For example, take an example of rat. It converts up to 50% of fructose to lactic acid as measured by the mesenteric vein cannulation. But in case of pig, it produces very little. So fructose in the blood of mature animals is very low, but in fetal and newborn lambs and pigs, it's very high. Sometimes the monosaccharides, they are not converted to glucose in the intestinal mucosal cells. So how these metabolic conversions take place is that the absorption may be converted to glucose by reactions in the liver. The animal body stores very little energy as carbohydrates, but some glucose is converted to glycogen. This glycogen is stored in liver and muscle tissues. What this glycogen is actually, it's a starch-like compound that can be hydrolyzed rapidly back to glucose. Thus, we can say that the level of blood glucose is maintained within a narrow range of normal animals by conversion of this circulating blood glucose to glycogen, and this process is called as glycogenolysis. So what actually glycogenesis is the conversion of this blood glucose to glycogen. This process is called as glycogenolysis. The blood glucose concentration increases after a meal, but returns to the fasting level within a few hours. Storage of glycogen after a carbohydrate meal prevents marked elevation in blood sugar and release of glucose by breakdown of glycogen. It has been found that the formation of glycogen from glucose, that process we call as glycogens, requires two molecules of ATP, that's the adenosine triphosphate, for every molecule of glucose. So glucose molecules that are added 1 unit at a time to form the long chain of glycogen, we need 2 molecules of ATPs, that's the adenosine triphosphate. Phosphorus uptake by adenosine diphosphate to form ATP, that process is called as oxidative phosphorylation. So this oxidative phosphorylation is actually a driving force for any biochemical processes, including the absorption of nutrients from the gastrointestinal tract. And sometimes it helps in the synthesis of proteins, nucleic acids, fats, and carbohydrates. The amino acids that are used for gluconeogenesis. What the gluconeogenesis is actually? The glucose that can be formed by body tissues, primarily liver and to a lesser extent kidneys, from non-carbohydrate metabolites, including lipids and amino acids. That's called as gluconeogenesis. These amino acids that are used for gluconeogenesis or for energy enter the citric acid cycle. This citric acid cycle is also called as Krebs cycle or the TCA cycle, tricarboxylic acid cycle. So it has three names. Amino acids not used for protein synthesis enter the general pool of metabolites that provide energy for normal body maintenance and productive functions. The important difference exists between the ruminants and non-ruminants in the importance of gluconeogenesis in energy balance. Ruminants, through fermentation of their digesta by microbes residing in the rumen, constantly depend on gluconeogenesis to meet their glucose needs. The glycogen storage is limited, therefore, when ingestion of carbohydrate exceeds the current needs for glycogen formation, the glucose is converted to fat. This conversion is accomplished by the breakdown of glucose to pyruvate. So there are different steps that are involved in this Krebs cycle. We will go through them step by step. So step one is the formation of citrate. The oxaloacetate condenses with the acetyl coenzyme A to form the citrate, which is catalyzed by the enzyme citrate synthase. So there are different kinds of enzymes that are involved in this tricarboxylic acid cycle, or we can say the Krebs cycle or the citric acid cycle. So these are the three names for this Krebs cycle. So first step, like I said, is the formation of citrate. In that the oxaloacetate condenses with the acetyl coenzyme A to form the citrate. And this reaction is catalyzed, or we can say is increased, the rate of the reaction is increased by the enzyme citrate synthase and inhibited by the adenosine triphosphate or nicotinamide dinucleotide citrate. These are the competitive inhibitors of oxaloacetate. The citrate that's formed in the second step is isomerized to isocitrate by the enzyme aconitase. So this is achieved by a two-stage reaction of dehydration followed by the hydration through the formation of intermediate cis-aconisase. The isocitrate dehydrogenase, that's the enzyme that catalyzes the conversion of isocitrate to oxalosuccinate. So like you can see in the figure, we are here, the citrate is the first step, then the isocitrate is formed, and then after that, the isocitrate dehydrogenase catalyzes the conversion of this isocitrate to oxalosuccinate. And then it is converted to alpha ketoglutarate. The formation of NADH and the liberation of carbon dioxide also occur at this stage. This reaction is stimulated by the isocitrate, the NADH, and magnesium dipositive, adenosine diphosphate, calcium dipositive links with contraction, and are inhibited by the NADH and ATPs. The succinyl coenzyme A that's formed in this Krebs cycle is converted to succinate by the succinate thiokinase. So you can see here that every step of the Krebs cycle reaction, it requires an enzyme in order to increase the rate of reaction or catalyze the reaction. So this succinyl coenzyme A that's formed in the Krebs cycle is further converted to succinate by the succinate thiokinase enzyme. The reaction is coupled with the phosphorylation of GDP to GTP. So this is a substrate level of phosphorylation. The glucose triphosphate is converted to ATP by the enzyme nucleoside diphosphate kinase. So the succinate that's formed is oxidized by the succinate dehydrogenase to fumarate. So that's the next step in the Krebs cycle. The fumarate is further converted to the malate. So that's the other step of the Krebs cycle. The formation of malate, the enzyme fumarase catalyzes the conversion of this fumarate to malate with the addition of water. The malate that's formed in this Krebs cycle is then oxidized to oxaloacetate by malate dehydrogenase. So the final synthesis of NADH occurs at this stage. The oxaloacetate that's formed is regenerated, which can combine with L molecules of acetyl coenzyme and continue the cycle. So that's the Krebs cycle, how it occurs. So these are different steps that are involved in this cycle. So let's talk about the diabetes mellitus. What is actually diabetes mellitus? So diabetes mellitus is relatively common in humans, but apparently it occurs less frequently in other animals too. So the disease is actually diagnosed partly on the basis of higher than normal fasting levels of blood glucose. This disease is not a single entity, instead it's a spectrum of clinical disorders of which excessive blood glucose, or we can say this excessive blood glucose is also called as hyperglycemia, which is a common denominator. So we have three basic types of or insulin dependent diabetes, mellitus, also called a juvenile onset or type 1 diabetes. And we have non-insulin dependent diabetes, also called as adult onset or type 2 diabetes. And then we have one more kind of diabetes, that's the gestational diabetes. So this type 1 diabetes and type 2 diabetes and the gestational diabetes, they are the three basic types that are insulin dependent diabetes or insulin independent diabetes or the gestational diabetes. The common metabolic defect in each of these forms is that there is improper production or utilization of the hormone insulin. You all might be knowing that this insulin is secreted by the pancreas. So the improper production and utilization of the hormone insulin secreted by the pancreas actually leads to these three types of these diabetes. Talking about the gestational diabetes, it's induced by increasing tissue resistance to the action of insulin and involves an increase in blood glucose and blood ketones. Gestational diabetes is probably the most common form of disease in domestic animals. Because insulin is required for normal glucose utilization, so its absence or cellular resistance to it results in hyperglycemia. Or it can lead to the urinary loss of glucose, that's called as glycosuria, and other changes as described for the ketosis. So here in this figure, you can see the glucose tolerance curves in the normal, diabetic, and hyperinsulin human. So this is the normal, diabetic, and hyperinsulin human. You can see here. So hyperinsulin human are shown in this figure. So what a glucose tolerance test indicates that not only the ability of the pancreas to secrete the insulin, but also ability of the liver to utilize glucose. Mild diabetes mellitus can be partially controlled by feeding low carbohydrate or high protein diets. Sometimes the diabetes can be induced artificially by administration of afloxacin. That's a drug that destroys the pancreatic cells that produce the insulin. Protein and amino acid metabolism, as well as carbohydrate and lipid metabolism, are affected by the insulin deficiency. So what happens with the insulin deficiency is that it results in severe negative nitrogen balance. There is glycosuria, that's the glucose loss in urine. excessive urine volume, or we can say the polyuria, and electrolyte loss associated with hyperglycemia and ketonemia. So we can say that carbohydrates, they are the major components of the plant tissues, and plants synthesize the carbohydrates using solar energy, carbon dioxide, water, and they give off oxygen. So this, you all know that that's called the process of photosynthesis, which is a vital for animals, and they could not exist without this transformation. Carbohydrates contain the carbon, hydrogen, and oxygen in a ratio of 1:2:1. The simplest carbohydrate is glucose, which is a monosaccharide such as and contains the 6 carbon atoms. A disaccharide, we all know that has 2 molecules of a simple sugar, for example, the table sugar or sucrose composed of glucose and fructose. An oligosaccharide has 3 to 10 sugar units and a polysaccharide over 10 sugar units. It has been found that animal tissues, they contain only trace amounts of carbohydrates. The plant tissue contains the mono, di, and polysaccharides. Polysaccharides include the starches that are hydrolyzed by digestive enzymes of animals and cellulose. Sometimes it has been found that lignification increases with plant age. It's a major structural component of mature plants and of trees. The amount of lignin that's present affects the cellulose and hemicellulose bioavailability of microbes, inhibiting the animal digestive tract, and in this way affects the nutrient value of the plant materials. There are other plant fibers also that include the gums, the beta-glucans, and the glycans. Soluble carbohydrates that are ingested by non-ruminants are hydrolyzed in the small intestine and absorbed by the active transport as monosaccharides. The glucose can be formed in body tissues from lipids and amino acids. We talked about the gluconeogenesis process. Absorbed glucose and that resulting from gluconeogenesis can be stored in limited amounts as liver and muscle glycogen, or that can be converted to body fat by breakdown to pyruvate and then enter the Krebs cycle. The complex cascade reactions by which glucose and its metabolites are oxidized or to provide energy is called as Krebs cycle. We talked about that in our last slides. So it has been found that the total energy that's released in the conversion of glucose to carbon dioxide is 673 kilocalories per mole. Sometimes the abnormal carbohydrate metabolism in animals occurs that's associated with diabetes and ketosis. We already talked about the diabetes that's associated with the elevated blood sugar level or blood glucose that results from the insufficiency of insulin from the pancreas or defective cellular sensitivity to insulin. So that was all about the carbohydrates. So moving to our next chapter which is macro mineral elements. So we will talk about certain kind of elements that are essential for the well functioning of the animal body. So these are the 7 macro mineral elements that are required by the animals to carry on their the life processes. So it has been actually shown that there are at least 22 inorganic elements that are required by some animal species and an additional 5 or more elements that may be essential. So the required elements can be divided into 2 groups. We have the macro elements and the micro elements. So that's based on the relative amount that's needed in the diet. For example, macro elements. The macro mineral elements include the calcium, chloride, magnesium, potassium, sulfur, sodium, and phosphorus. So these are the macro mineral elements. So some macro minerals such as calcium, magnesium, and phosphorus, they are regarded as structural components of the skeleton, while as others such as sodium, potassium, and chlorine, they function in acid-base balance. Talking about the magnesium being a constituent of bone, it's required for activation of the several enzymes. So all macrominerals have one or the other functions. So we will talk about each of the macromineral element, their functions, the deficiency symptoms, or some toxicity levels in our coming up slides. So starting with the calcium, which is the most abundant mineral in the body. It has been found that 99% is found in the skeleton. So calcium supports muscles and protects the organs and tissues, including the bone marrow. It's required for growth and milk production. The calcium requirement decreases with age, but increases with the growth. So requirements for ewes rise rapidly during late pregnancy to a level equal to that for lactation. So for goats, calcium requirements are higher during pregnancy, but they increase abruptly after killing. Calcium is generally not regarded as a toxic element because excess calcium is excreted in the feces. So according to the National Resource Council, the maximum dietary level of calcium is 1.5%. So prolonged calcium deprivation can manifest itself in a variety of ways depending upon the stage of growth. So functions of calcium, there are four main functions of calcium. That's skeletal structure, cellular metabolism, the bone formation, and excretion. So let's talk about them one by one. So skeletal structure. So how the movement of calcium to and from the skeleton occurs. So this calcium, it plays an important role in regulating circulating concentration of ionic calcium and thus calcium absorption. So around 1/5 of calcium in the skeleton is mobilized in sheep and dairy cows in association with parturition and the onset of lactation. The resorption process is probably the obligatory since it's not abated by supplying more dietary or protein and is not necessarily accompanied by the hypocalcemia. 99% of calcium is found in the skeleton, so a strong framework for supporting the muscles and protects the delicate organs and tissues. That's the major function of the calcium. The skeletal reserve of calcium actively supports the calcium homeostasis in what Bain and Watkins described as an elegant compromise. So sometimes the bone apposition occurs through the activity of osteoblasts. So these are the bone forming cells at the growth plates and in other areas of rapid bone growth. This bone resorption occurs by two processes. One is by resorption of bone surfaces by the activity of osteoclasts, and the other is the resorption deep within formed bone. That's the osteocytic resorption. So the later one, the osteocytic resorption, is generally believed to be the major process of bone resorption. The continuous apposition and resorption of bone, we might call it as bone remodeling, is the basis for bone growth and for changes in its shape and density. So this continuous process of bone remodeling results from simultaneous bone apposition and bone resorption that regulates the bone growth in the young animals and bone homeostasis in the adult animals under certain physiological conditions like pregnancy, lactation, or maintenance. So some more functions of these, the calcium is that nuclear receptors for 1,2,5-OH2D3 on the chondrocytes and osteoblasts. That's the dihydroxycholecalciferol, we can call it. The receptors for the PTH are found linked to regulatory, that's the parathyroid hormone, the PTH. They are linked to regulatory G proteins in the organs such as kidneys and brains. Calcium accretion and resorption are coupled during growth and uncoupled in late pregnancy and early lactation. So these parathyroid hormones are linked with the proteins and then enter the maternal circulating and mimic the parathyroid hormone. So we already talked about the bone resorbing cells, that's the osteoclasts. They respond to the 1,2,5-OH2D3, that's the dihydroxycalciferol, we can call it. These bone resorbing cells respond to these cytokines released by the osteoblasts and to estradiol, increasing insensitivity to parathyroid hormone that's linked with the proteins. Calcitonin, we all know that, is now believed to have an important role in limiting the bone resorption during lactation, but this doesn't prevent the growth regulation of calcium absorption. So let's now talk about the other function of calcium, that's the cellular metabolism. So calcium controls the excitability of nerve and muscles. So reduced calcium dipositive ion concentration produced increases excitability of pre and post ganglionic nerve fibers. So if we have higher than normal calcium dipositive ion concentration, they have the opposite effect on nerves and muscles. Calcium is also required for normal blood coagulation. That's one of the cellular metabolism functions of calcium. So the great importance of calcium in cellular metabolism has recently become appreciated through knowledge of its role in sarcoplasmic reticular function in skeletal and cardiac muscles, and also in the physiological functions of the nervous system. So overall movement of calcium in metabolism is shown in this figure here. So in the growing animals, net retention of calcium occurs in the body. That's the amount stored in bone and other tissue exceeds that lost in feces, urine, and sweat. So in non-pregnant or non-lactating adults, the amount of calcium ingested is equal to the amount lost if the metabolic requirement is met. So like you can see in this figure, the endogenous fraction is reabsorbed partially, as you can see in the figure. So therefore that appearing in the feces is termed fecal endogenous calcium. So this is the fecal endogenous calcium you can see here in the figure. So this is the dietary calcium, that's the calcium intake. The apparent calcium absorbability, or we can say the feed calcium minus the fecal calcium, generally approximates about 50% through the percentage, although the percentage tends to decline as the intake increases. So we also have the urinary output of calcium here. It's less than that of the fecal output in most species. So about half of the plasma calcium that's mainly ionized calcium is filtered in the kidney, and more than 90% of this amount is reabsorbed under normal conditions. Loss of calcium in sweat is of only minor significance in most cases, but in humans, horses, and other species in which sweating is prominent, large amounts of calcium can be lost by this route. The sweat loss of more than 1 gram of calcium per day occurs in adult men doing heavy physical work at high environmental temperatures. Sweat losses sometimes equals or may exceed the fecal and urinary losses in these men. So urinary excretion tends to remain low and constant regardless of calcium status, although it may rise significantly in the acidotic dairy cow. So there are a lot of deficiencies that occur because of the calcium deficiency. So deficiency symptoms that occur because of the deficiency of calcium. So there are some abnormalities of bones. We might call it as rickets. The type of bone disorder that develops is partly dependent on the age at which the calcium deprivation develops. Talking about the rickets, that's the abnormality of bones, that's one of the symptoms of calcium deficiency. It's a non-specific term used to denote the skeletal changes and deformities in young animals, and it's characterized by a uniform widening of epiphyseal or diaphyseal cartilage and an excess amount of uncalcified tissues, causing the enlargement of ends of the long bones and of rib costochondral junction. You can see like here, they are like the legs of the pig here, they are like twisted. So it actually changes the skeletal formation or causes some deformities in the skeletal structure. One more is the osteomalacia. It describes the excessive mobilization of the minerals, leaving a surfeit or matrix of bones in which the growth plate has closed. So incomplete mineralization of osteoid or osteoid in adults, which is called as osteoporosis. That indicates that bones contain less mineral than normal, but proportionately less matrix. So the degree of mineralization of matrix remains the normal. This matrix osteoporosis is the characteristic of protein deficiency rather than simple calcium or phosphorus deficiency can occur in young animals as well as the mature animals. Deficiency of calcium, sometimes in presence of excess phosphorus, may also cause abnormal bone. But in the instance, excess bone resorption by osteocytic osteolysis results in fibrous osteodystrophy. So what this looks like is shown in the picture here. So sometimes the deficiency of calcium in presence of excess amounts of P causes some abnormal bone deformities. This condition is characterized by the replacement of osseous tissue with fibrous connective tissue. So like here in this picture, you can see the nutritional secondary hyperparathyroidism in a horse, a big head here. The facial bones are enlarged and the facial crest is no longer visible. So at the bottom here, you can see some radiographs of a section of metacarpus of horse, which is fed a normal calcium is to phosphorus diet, and on the right horse fed excess P. So in the nutritional secondary hyperparathyroidism, the cortex is thinner and radiographic density is markedly decreased. Also, you can see here a pig with a distorted snout that's caused by the nutritional secondary hyperparathyroidism that was induced by a dietary calcium phosphorus imbalance. That means even though there is deficiency of calcium, but excess amount of P, that's the calcium phosphorus ratio, that results in the distorted snout of a pig because of the nutritional secondary hyperparathyroidism. The calcium deficiency may also cause the calcium tetany, that's called the milk fever in grazing herds. Older cows and those needing assistance at calving had an increased risk of developing this milk fever. So this calcium tetany is also called as milk fever. So high alkalinity or dietary cation or anion difference due largely to high pasteurization or potassium may increase the risk of milk fever. So there is a tendency for hypocalcemia to be associated with the high urinary pH. So this high potassium concentration in the conserved forage lead to raised DCAD rations fed to the housed herds. So severe calcium deficiency, or we can say the milk fever that we talked about, or it's also called as calcium tetany, it may produce the hypocalcemia, which results in the tetany and convulsions. So pathogenesis of this calcium tetany in the animals is like calcium deficiency, deficient intake, or sometimes there's disturbance in absorption with vitamin D deficiency or diarrhea, or excessive kidney excretion. Or sometimes there are formation of complexes or parathyroid hypofunction. So moving to the calcium toxicity. So acute calcium toxicity has been reported, but chronic ingestion of calcium in excess of metabolic requirements results in abnormalities in bone, which can be regarded as manifestations of the toxicity. The tendency towards hypercalcemia resulting from continued absorption of excess calcium stimulates the calcitonin production by the thyroid. So this inhibitory effect of calcitonin on bone resorption is homeostatic mechanisms that's aimed at the minimizing the sources of serum calcium. So here in this picture you can see the excess bone mass, that's the osteoporosis in dog fed excess calcium that's on the top, and the this one is the normal diet, that's the normal calcium fed dog. So second most important macromineral is the phosphorus. We all know that the phosphorus content of adult humans approximates about 1.1% of the fat-free body, of which about 80% is in the skeleton. So if we take the bone ash, it contains about 18% of phosphorus. The percentage of phosphorus in the body and the proportion of the total phosphorus in the skeleton increases throughout the perinatal and postnatal life. The phosphorus constitutes about 22% of the mineral ash that we already talked about. The calcium and phosphorus we talked about, they are closely associated with each other in animal metabolism. So adequate calcium and phosphorus nutrition depends on three factors. One is the sufficient supply of each nutrient, a suitable ratio between calcium and phosphorus, and the other is the presence of vitamin D. So these factors are actually interrelated. So the desirable calcium phosphorus ratio is often between 2:1 and 1:1. So as with calcium, the most important functions of calcium is it's a component of skeleton. So in this role, it provides the structural support for the body because calcium and phosphorus, as we already talked about, they occur together in the bone. So we already talked about that in the section of calcium. So talking about the function of P, it functions in energy metabolism as a component of adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate, and of creatine phosphate. The signs of a P deficiency, the most common sign of P deficiency in growing animals is rickets, as you can see in figure here, as it's closely related to the calcium. So the gross and histological change in rickets are described earlier. We already talked about that in the calcium section. The fecal output of phosphorus tends to remain relatively unchanged while the urinary excretion is reduced. But the total excretion may still exceed intake when the calcium intake is relatively high. So what we want to actually say here is that both the calcium and phosphorus ratio, they are interrelated. So calcium intake is excretion in both urine and feces is increased in P deficiency. That means if we have a P deficiency, then the calcium excretion in urine and feces is increased. So that results in certain kinds of deficiency symptoms. So as the deficiency progresses, the appetite fails and the growth is retarded. So deficient animals, as you can see here in the figure, often have a depraved appetite and may chew on wood or other inappropriate objects. So this abnormal behavior of eating or chewing is called as pica. So adults fed low phosphorus diet may exhibit pica. That means they might start chewing some wood or any other items, and bone density is decreased, as you can see in the ricket section here. So these are the P deficiency, like we talked about the pica. A P deficiency may result in a deprived appetite, so they start eating some wood or some other materials. So here in this figure are shown some of the material that are recovered from the stomach of a deficient cow. So it might include the oyster shells, the porcelain, the tire casing, pieces of metal, or some pebbles. So this abnormal behavior of the animals occur because of the P deficiency. So moving to the magnesium, it's distributed widely in the body, except for calcium and phosphorus that's present in larger amounts than any other minerals. So about half of the body magnesium is in bones at a concentration of about 0.5 to 0.7% of the bone ash. So magnesium in soft tissues is concentrated within the cells. The highest concentration is in liver and skeletal muscles. So the different functions of the magnesium are that it's required for normal skeletal development as a constituent of bone. It's also required for oxidative phosphorylation by the mitochondrion of heart muscles or mitochondrion of the other tissues. Magnesium also plays an important role in the activation of enzymes that split and transfer phosphatases and the many enzymes concerned in the reactions involving ATP. So as we have already discussed, the ATP that's required in such diverse functions as muscle contraction, protein, nucleic acid, fat, and coenzyme synthesis, or we can say the glucose utilization. So by interference, we mean the activation action of magnesium extends to all those functions. Magnesium metabolism is complex and varied. So absorption from the gastrointestinal tract occurs through the ileum. Magnesium deficiency in growing rats results in anorexia, reduced weight gain, or we can say the reduced serum magnesium. So we have some hypomagnesiemic tetany also that occurs because of the deficiency of magnesium. So recent evidences suggest that magnesium deficiency may be a causative factor in osteoporosis in animals and humans, but still some research is going on this, that whether that uh, claim is right or wrong. That means there are some evidences that suggest that magnesium deficiency may be a causative factor in osteoporosis in animals and humans. Common problem of grazing cattle that's because of the magnesium deficiency is called as grass tetany. Like we already talked about the calcium tetany because of the deficiency of calcium. So this is different. This is because of the deficiency of magnesium. So common problem of grazing cattle is a syndrome that's called as grass tetany, also called as grass staggers. Magnesium tetany or wheat pasture poisoning. It occurs most frequently in cattle that are grazing cereal forages or native pastures in periods of lush growth. But at times it may also be a problem in cattle that are fed the conventional wintering rations. So what are the symptoms of this grass tetany is that hypomagnesemia, that means the low blood magnesium. It has been found that the etiology of magnesium tetany is not understood completely, although it's generally agreed that hypomagnesemia, that means the low blood magnesium, hypomagnesemia is the low blood magnesium, is underlying the cause that triggers the factor. So it has been found that some scientists have come to the conclusion that high level of potassium and protein usually present in those green pastures, that might be the possibility of an antagonism with magnesium. So magnesium toxicity, talking about the magnesium toxicity in animals includes the depressed feces intake, feed intake, diarrhea, losses of reflexes, and cardiorespiratory depression. So sometimes because of the toxicity of magnesium, there might be severe diarrhea, and also the reduced feed intake and growth have been observed in calves that are fed diets containing more than 2.3% of magnesium. And accidental feeding of high magnesium diets to sheep results in the diarrhea and anorexia. So it has been found that intravenous injection of magnesium sulfate more than a century ago that resulted in the peripheral paralysis of muscle paralysis of the dogs. So next macroelements is the potassium, sodium, and chlorine. They are considered together because they're all electrolytes. They play a vital role in maintaining the osmotic pressure in the extracellular and intracellular fluids and in maintaining the acid-base balance. In addition to that, each of these has its own special functions. So total body contents of potassium, sodium, and chlorine in newborn and adults of several animals, including man, they are shown in the table in your book. So normal ratios among electrolytes are constant among species. So here is the diagram that shows the comparison of electrolyte concentration in plasma, interstitial fluid, and intracellular fluid. So the height of each column you can see here, it represents the total electrolyte concentration. Potassium is present mainly within cells. About 90% of body K is intracellular and is readily exchangeable with the extracellular fluid. On the other hand, sodium that's present largely in extracellular fluid with less than 10% within the cells. So of the other 90%, about half is adsorbed to the hydroxyl apatite crystal or bone, and half is present in the plasma and interstitial fluids. Chlorine, which acts with the bicarbonate to balance electrically the sodium of the extracellular fluid. So excess excretion of sodium by the kidney is accompanied by chlorine excretion. Chlorine is present almost exclusively in the extracellular fluid, as you can see here in the figure. The concentrations of these electrolytes and other constituents in the blood plasma, intracellular fluid, and intracellular are depicted. Intercellular and intracellular fluid are depicted in this picture or in this figure. The composition depicted for intracellular fluid is only an approximation because some individual tissues have cell electrolyte composition that's considerably different from the mean. So the main functions of K is it's located mostly within the cells and by means of an energy requiring system related to sodium movement, it influences the osmotic equilibrium. That's what maintains the osmotic, it acts as an osmotic gradient. So it's required in enzyme reactions involving phosphorylation of creatine, and it's required for activity of pyruvate kinase. The critical roles of sodium, calcium, and chlorine are in maintaining the acid-base balance, that's the homeostasis. The overall acid-base balance in the body is maintained by controlling the hydrogen ion concentration of body fluids. So talking about the metabolism of potassium, sodium, and chlorine ions, they are not absorbed in appreciable amounts from the stomach, but considerable absorption takes place from the upper small intestine and some lesser amounts from the lower small intestine and large intestine. Concentration of these potassium, sodium, and chlorine ions is regulated within rather narrow limits in extra and intracellular compartments of the body. So it has been found that ingestion of more of each mineral and its needed results in ready excretion by the kidneys. So toxicity is not likely unless water intake is restricted. So there are not any toxicity symptoms that we can see unless the water intake is restricted, which impedes the urine output. So deficiency symptoms of potassium results in abnormal electrocardiograms in most or all species. Sometimes there are kidney lesions, the growth retardation, and muscle weakness. Main signs of sodium deficiency are the reduced growth rate you can see and feed utilization in growing animals, and sometimes there is reduced milk production and weight loss in adults. Chlorine deficiency results in depressed growth rate. That seems to be the major sign of the chlorine deficiency. So low dietary chlorine results in a decline in urinary chlorine concentration to near zero, and sometimes the chlorine concentration of skin, muscle, liver, kidney, brain, and other tissues is also reduced. So like we already discussed that the kidney normally regulates its excretion of potassium, sodium, and chlorine in accordance with the variations in dietary intake. So therefore, a toxicity of any of these three electrolytes is unlikely except their water intake is restricted. So drinking water is saline that's high in these ions, or animals have kidney malfunction. So chronic K excess induced by one of the above means results in hypertrophy of the adrenal cortex. So this happens only if there is some limited water intake. So as such, there are no certain toxicity symptoms because of the sodium, chlorine, and potassium. So we have the last one macro mineral element, that's the sulfur. So sulfur helps in the tissue distribution. So animals require sulfur mainly as a component of organic compounds, including the amino acids, methionine, cysteine, or vitamin biotin and thiamine. Because proteins are present in every cell of the body, and sulfur containing amino acids are components of all proteins. So main functions of the sulfur are that sulfur mainly functions, its presence in organic metabolites. So inorganic sulfate from exogenous dietary sources and from endogenous release from sulfur containing amino acids are mostly utilized in synthesizing the chondroitin matrix of cartilage or in biosynthesis of taurine, the heparin, cysteine, and other organic constituents in the animal body. In birds, if we talk about the sulfate is incorporated into feathers, the gizzard lining, and muscles. So its functions in organic compounds will be listed in terms of the metabolic functions of the compounds that are contained in it. So talking about the metabolism of the sulfur, the absorption of inorganic sulfate from gastrointestinal tract is inefficient. Active transport of this sulfate takes place from the upper small intestine. So both inorganic and organic forms of sulfur are actually used for the sulfation of cartilage mucopolysaccharides. Some of the deficiency symptoms of sulfur are that inorganic sulfur has not shown to be essential for animals for normal maintenance or productive function. So deficiency of any of the organic metabolites that contain the sulfur produces some functional and morphological lesions. So toxicity of sulfur, the because the intestinal absorption of inorganic sulfur compounds is low, the sulfur toxicity is not a major problem. So however, a serious neurological disease, that's the polio, that's the polioencephalomalacia, PEM, can occur in ruminants that are fed high levels of sulfur, which leads to excessive production of sulfides in the rumen. So talking in brief about the summary, what we talked about, the macro elements. So required macro minerals are the calcium, magnesium, phosphorus, sodium, chlorine, potassium, and sulfur. Calcium, magnesium, and phosphorus are required as structural components of the skin. The calcium deficiency results in reduced bone calcification. We talked about the rickets. We also talked about the phosphorus, which is important in lipid transport and cell membrane structure. We talked about the magnesium that functions as a skeletal constituent and is required for oxidative phosphorylation by mitochondria of heart muscles or any other tissues. Sodium and chlorine, they are the electrolytes that are vital in maintaining osmotic pressure in intracellular and extracellular fluids. Potassium is vital in maintaining osmotic pressure and acid-base balance. We talked about sulfur that's required mainly as a constituent of organic metabolites. In such, there are no certain deficiency symptoms unless it's related to those organic metabolites. There are not certain toxicities also related to the sulfur, but sometimes there might be some serious neurological disease like like we may call it polioencephalomalacia, PEM, that can occur in ruminants which are fed high levels of sulfur. So interactions between and among the macrominerals and macrominerals and between macrominerals and microminerals are increasingly recognized as important in nutrition. So that was all about the macrominerals. So moving to our third topic is the mineral toxicities and organic toxins that are present in the food chain. We all know that all stable and some radioactive elements in the periodic table are present in the Earth's crust. So unlike organic compounds whose presence in nature depend on synthesis by living organisms, the mineral elements are finite and are not distributed uniformly on the planet. So translocation of these minerals through human intervention, for example, the mining, agriculture, physical disturbances like glaciers, earthquakes, hurricanes, or volcanic eruptions, they have led to the differences in geographic distribution and concentration of these minerals. In addition to the mineral toxicities, many organic toxins are encountered by the animals and humans in the feeds or food. For example, we have here heavy metals, metals having atomic weight greater than sodium and specific gravity greater than 5 grams per cubic centimeter. So on health effect basis, we have some essential, non-essential, and less toxic and highly toxic. So lead, mercury, and cadmium, they are considered as highly toxic. Stannic and aluminum, they are considered as less toxic, while as beryllium, barium, lithium, and zircon, they are non-essential. So mineral toxicity is actually because of all stable and some radioactive elements in the periodic table that are present in the Earth's crust. So we already talked about the translocation of minerals, how these are translocated because of mining and agriculture, and because of the physical disturbances, for example, glaciers, earthquakes, or hurricanes that contributed to the differences in geographic distribution. Sometimes, in addition to mineral toxicities, many organic toxins are encountered by animals and humans that use the food or feed for the animals. So these are the different sources of mineral toxicity. So talking about the enigma of mineral toxicity, all inorganic elements, indeed all nutrients, may be toxic to animals when ingested in the excess amounts. So the margin of safety between the minimal amount required in the diet and the amount that produced adverse effects varies among minerals and according to conditions. So there are certain limits for that. Or we can say the maximum tolerable levels of minerals for dietary animals. So there is a table in your book that represents certain maximal tolerable levels of dietary minerals for domestic animals. Beyond that level, they might be toxic for the animals and produce some toxicity symptoms. So here we will discuss some of the minerals in that present more pressing practical problems for humans and animals. For example, we have lead, cadmium, mercury, fluoride, and molybdenum that are inexorably linked to animal nutrition. So starting with the lead, so sources of lead in the environment are like metal plating and finishing operations, sometimes the waste from the battery industries, we have soil wastes, the exhausts from the automobiles and addition in gasoline and pigment, sometimes the smelting of ores that result in the lead pollution in the environment. We have some fertilizers and pesticides, the factory chimneys that release a lot of lead in the environment. So lead poisoning was actually recognized several hundred years ago and became a clinical problem in Western Europe in the 17th and 18th century, where cooking utensils, the household articles, and water pipes contained high concentration of lead. So in more recent times, it has been found that lead poisoning in children become more common as a result of ingesting residues of the high lead paint or eating and drinking from utensils made of high lead material. So, chronic lead poisoning in people of all ages is a threat whenever the water supply passes through tanks and pipes that are covered or coated with lead. Lead toxicity presently is considered the most common cause of accidental death by poisoning in human and animal worldwide. So, signs of lead toxicity include the pallor, lassitude, anorexia, and irritability in both children and adults. But the most definitive symptoms are anemia, elevated blood lead levels, and urinary excretion of alpha amino levulinic acid in humans and animals. The lead toxicity actually affects a large number of organs and tissues. Some nephrological, endocrine, reproductive, or gastrointestinal tract, or hepatological or neurological. So, microcytic hyperchromic anemia is produced in lead toxicity as a result of decrease in survival time of red blood cells and a decrease in red cell formation from a block in hemosynthesis. So effects of lead toxicity is the necrosis, hemorrhage, and ulceration of stomach and small intestine that occur in animals. So in the kidney, all pathological changes that occur result in aminoaciduria or glycosuria and hyperphosphaturia. Lead toxicity also affects the skeleton, causing osteoporosis and reduced bone matrix formation. Enlarged joints of long bones are common in swine and horses. So moving to the cadmium, which is next to lead, the cadmium is toxic to a wide range of animal life and has adverse effects on the testes and kidney. Cadmium metabolism has been reviewed by National Research Council. It occurs geologically in zinc ores and is used widely in industry in the production of batteries, pigments, stabilizers, and in electroplating. The atmosphere around us have some states of United States contains significant amount of cadmium as high as 6.2 milligrams per cubic meter of cadmium and is a significant source of water pollution. Food contamination with cadmium is also variable among food resources. So effects of cadmium toxicity is that specific effects of cadmium and fat to experimental animals include kidney damage and hypertension as well as hypochromic microcytic anemia. So in animals, there is chronic ingestion of cadmium causes, if there is chronic ingestion of cadmium, it causes increased systolic blood pressure in the absence of significant renal disease. Such pressure effects have been linked to depressed blood and tissue levels of atrial uh, peptide, increased blood levels of aldosterone, and retention of sodium and water. So these are the different organ systems that are affected because of the cadmium toxicity is the kidney, respiratory system, the reproductive system. It causes the testicular necrosis, estrogen-like effects. That means the affection of steroid hormone synthesis. And when the cadmium toxicity as a skeletal system, when it affects the skeletal system, it causes the loss of bone density and mineralization. So talking about the effect of cadmium toxicity on kidneys, it causes the kidney stones or proteinuria or glomerular and tubular damage. And when it affects the respiratory system, it causes pneumonia or pneumonitis or destruction of mucous membranes. So these are the different toxic doses of cadmium in cattle and sheep. A series of unrelated compounds protect the testes against injection. These include the zinc, selenium, cysteine, glutathione, and estrogen. It has been suggested that selenium may protect by forming relatively unstable selenium salts of cadmium that are stored harmlessly in the body. So a lot of research has been done or is being carried out. So one of the research is that it is suggested that the selenium may protect this cadmium toxicity by forming unstable selenium salts of cadmium that are stored harmlessly in the body, so they don't present any harm to the body. So we have the mercury. How does mercury get into the environment? Is because of the global and regional resources, atmospheric transport and deposition, mercury pollution. Uh, because of the industrial pollution, also mercury levels have been increased in the environment. So effects of mercury toxicity is that it combines with SH groups, and in this way it inhibits the enzyme systems containing such groups. Mercury accumulates in the lysosomes within cells and has been associated with their rupture, resulting in hydrolytic enzyme release. Mercury poisoning also produces the kidney necrosis and death. Some organic mercury compounds such as methyl mercury and phenyl mercury are more toxic than inorganic mercury compounds. So all of these compounds cause actually the accumulation of mercury in liver and kidneys. So severe growth depression in rats and decreased egg production and hatchability is found in animals. So however, vitamin E protects against the toxic effect of this methyl mercury in mammals and birds. So here are some scenarios like a Florida panther was found dead with acute mercury toxicity in Florida. So compared to other mammals, this highly endangered species feeds at a very high level of food chain and continues to be at a risk of mercury exposure. So this Florida panther is actually an endangered species, and because of this ingestion of high levels of mercury. The the river otters, that means is an endangered mammal that inhabits in the rivers and streams across the US. So they are playful, energetic, that feed primarily in fish, but also feed on crayfish and other aquatic prey. So as a result, these otters also are taking a steady diet of mercury laden food throughout their lives. So mercury levels in these otters have been found in New England, so New York, Georgia, and Ontario. Their high mercury levels have been found. So talking about the fluoride toxicity in animals, the fluoride, aside from its role in preventing dental care, so we always use the toothpaste that's rich in fluoride. That means it should have fluoride, but sometimes the excess amount of fluoride is too dangerous for us. It might be toxic. So aside from its role in preventing dental care, the fluorine can be considered as a toxic mineral for humans and animals. The tissue distribution, metabolism, and toxic effects of fluorine have been described in detail in the National Research Council in 1971. So chronic fluorosis has been reported from several parts of the world as a result of fluorine-containing dusts from steel mills and other industries that are processing the fluorine-rich substances, such as the reduction of aluminum ore, and secondarily in association with the use of high fluorine rock phosphate as mineral supplements for animals. So the effects of high fluorine on bone are somewhat contradictory. Reported effects of fluorosis in animals range from osteoclarosis to osteoporosis. Methods for the control of fluorine involved are known and are not difficult of application, but the fluorine hazards for factory workers, for their families living nearby, and other people residents or employed in the neighborhood as well as for the animal population are not sufficiently appreciated. Fluorine actually is a potent enzyme inhibitor. So like I said, that fluorine, you have a great smile, but excess amount of this fluorine might be very dangerous or toxic, or it might produce some uh, diseases, or it may lead to even death if you ingest more fluoride. So molybdenum is the last micromineral that's required by the animals, or that uh, uh, the toxic levels of these in the food and feed chain provide certain symptoms. So although molybdenum is recognized as a required nutrient, its more concern is toxic minerals. So molybdenum toxicity was first recognized in cattle grazing. Certain pastures that are high in molybdenum develops severe diarrhea. The syndrome became known as Pete's scours or teeth, and did, and later was found to be prevented by copper supplementation of the diet. So subsequently, it was discovered that the high molybdenum and sulfate intake produced the copper deficiency in ruminants. Thus, the primary toxic effect of molybdenum intake is the precipitation of copper deficiency. Leguminous plants sometimes contain high molybdenum. Spring pasture contain high molybdenum than the autumn ones. So clinical symptoms of the molybdenum poisoning is that anorexia, immunosuppression, persistent diarrhea, dodgy hair, hind limb, or incoordination. So common sources of these excess copper or errors in feed formulation, consumption of feedstuffs formulated for other animals, grazing on pastures that are fertilized with pig or poultry manure, grazing forages deficient in molybdenum, use of copper containing anthelmintics, or use of copper containing food baths or copper supplements. So treatment is that usually unrewarding for severely affected animals, but administered both molybdenum and sulfate as a drench or added to the feed like ammonium molybdate or sodium sulfate. Reduce or eliminate the extraneous sources of copper. So many mineral elements are ubiquitous in the environment but have no known physiological requirement for animals, yet they enter the food chain. The trace minerals cadmium, fluorine, mercury, and molybdenum represent the practical problems of toxicity. Pollution of the environment with trace elements, both required, that's carbon and selenium, and not required, that's lead, mercury, and cadmium, and macrominerals, is of great importance. So many different poisonous plants are recognized, and large number of animals are affected adversely by the consumption of these plants. So contamination of cereal grains, the forages, and seed legumes produce mycotoxins that are toxic to livestock and humans. So that's all about the three topics that we covered right now. So if you have any questions, you can just